Good morning, everyone. How is everyone this morning? I see we have our panelists have arrived, and they're all looking bright and shiny, and you've all got your cups of coffee. We're really excited for today's event. We're going to have our first Alberta Pulse Growers Tailgate Talk. So we're going to give it another minute or two until we have all of our attendees logging on. We see you guys are coming in quick. For everyone who's just signing on, my name is Nevin Rosaasen. I'm the Policy and Program Specialist for Alberta Pulse Growers. I'm also a farmer from Eastern Saskatchewan, and on our farm, we grow a lot of different pulses. We grow peas, we grow lots of faba beans. We've even tried dry beans out in our area of the world. Again, I'm very happy to be here today facilitating. With Alberta Pulse Growers, I do agronomy and I also do policy. And before we introduce our panelists, I would also like to extend a very sincere thank you to the people behind the scenes. We can't make this work without a team of individuals, so I would like to thank Dr. Jen Walker, who is here on the webinars today or at the Tailgate Talk, Dr. Jagrup Gill-Kaloon as well. They take care of our research portfolios here at Alberta Pulse, and also to Rachel Peterson, our communication coordinator. So everyone, today it's my pleasure to introduce our four guests for, again, the APG Tailgate Talk. We have Syama, we have Holly, and we have Mike. And I'm going to give a little bit of an introduction to them before they kick things off and talk in their areas of expertise. But before that, we'd like to start a poll just to get a little bit, know a little bit more about you, the audience, etc. And I'd also welcome you to put in the questions where you're from and where you're attending today. So just would like to launch that first poll. If you could, Jagrup, that would be great. We're going to get an idea of what pulse crops are in your current rotation. So I'd invite you to answer that poll. We're going to give it a few minutes. We've got votes coming in already, and then we're going to close it. So again, this morning, this tailgate talk is going to be focusing on Aphanomyces euteiches and other root rots. And we're going to talk about which crops are, of course, susceptible, which ones are a little more resistant, and which ones do not have infection rates. So we've got 10 seconds left on this poll if you haven't yet voted, and we're going to close it up. And we're going to move on to the second poll. And again, I would welcome everyone to put in the question box where you're joining us from today. All right, if we could close that poll, let's see what the answer is. Let's bring it up. It looks like only 61% of you voted. You guys better top up your coffees here. That wasn't a very difficult task. So it looks like 45% of the attendees grow peas, 9% grow lentils, 18% grow faba beans, and 27% grow other, or a combination of those two. Excellent. All right, so we're also going to poll you one more time, and this is to get to know a little bit more about you, the audience, as we have our last few attendees trailing in here. So Jagrup, if you could open that second poll. The question is, I think I can bring up the question. Yes, here we go. Is Aphanomyces a problem in your area? So again, we invite you to answer yes or no. Perhaps it's root rots. Of course, we're going to get into the specifics on how you know if it is for sure 100% Aphanomyces as well. So while that poll is closing, I'm going to introduce our first panelist. Good morning, Syama. How are you today?
Good morning, Nevin. I'm good. I just had to take myself off mute.
Yeah. Today's Zoom meetings are modern day seances. It's like we're calling spirits from the other realm. So anyways, Syama joined Agriculture and Agri-Food Canada at the Lethbridge Research and Development Centre in 2011 as the pulse and special crops pathologist. Her research focus is primarily on management of root diseases of pulse crops and molecular diagnostics and identification of soil-borne pathogens. She published the first report describing Aphanomyces root rot in pea in Alberta in 2013 and has been on a journey of discovery of this disease ever since. She is keen on bridging molecular lab tools with real in-the-field solutions and believes that knowing what pathogen and how much is in your field is the first step to management. Besides her passion for pulse diseases, she has also a passion for cooking with pulses, and particularly trying all sorts of different tricks to get her kids to eat pulses, something that I'm also working on as well. So Syama, I'm just going to bring up your presentation here, and then we'll ask you to take it away. All right, the floor is yours.
Okay, thank you, Nevin. I can't tell if my screen is up or not. I'm still seeing the results of the poll. There we go. Okay, good morning, everyone. I'm happy to be here this morning to talk about my favorite topic, root rots of pulses. And this morning, I think, because this is a little more of an informal session, I've gone about this from a different way. And I thought what I would focus on was looking at kind of the retrospective. So what have we done for root rot research in the past 7 years or so? And then what are the prospectives for root rot research? So what do we do to what do we need to do to move research ahead? So a little less of a science talk, because at this point, figure most people know Aphanomyces and know all the different players. And so I wanted to look more at kind of high level and what the research, where the research has been and where it's going. So next slide, please. So I've spent, the last few months, we're trying to develop a root rot research working group, quite a mouthful. So I spent the last few months trying to develop what the research vision for root rot management should be and how we can bring that all together with lots of different collaborators. And as Nevin said in my bio, really, I think that, the foundation for management is a soil test, or knowing what's in your soil, so knowing what you're dealing with. And in order to do that, we need reliable pathogen quantification. So that's a big part of what my research is. And we need to have a reliable pathogen quantification, not just for fields that are very heavily infested, where you would suspect you already have a problem, but for fields where we're starting to see new infections coming in and starting to see patches. Because I think, looking towards the future, the biggest thing that we can do is prevent those patches from spreading to become a big problem throughout the field. So starting to focus more on kind of suboptimal threshold levels for quantification and looking at how can we prevent these patches from spreading. So once we know what's in the soil, then the next step would be managing diseases, of course, and that's what our ultimate goal is. And so there's a lot of different questions around that. And I would say, as I go through this presentation, we'll see that managing disease is really the biggest challenge. So some of the things that we're looking at is that interval period. So how long do we need between pulse crops to either manage the disease or make sure the disease doesn't take off in the first place? How are these oospores or the inoculum of Aphanomyces, how is it surviving in the soil and how long does it survive in the soil? And then, the other sort of general agronomic things like, are they crop rotations that can help? There are other practices like seed treatments, other in-crop practices that can help. And then ultimately the goal is also to have partially resistant cultivars as a method to manage diseases. And that's also a challenge for this pathogen. And once partially resistant cultivars are released, hopefully sometime in the near future, we have to manage those resistant cultivars so we don't lose that resistance. So we have to know what the threshold level is going to be for partially resistant cultivars. I don't think we're going to be able to deploy them in very heavily infested fields because we don't want that resistance overcome. And then there's also just a continual challenge of finding new resistant sources, developing new germplasm, so we're not relying on the same resistance. Next slide, please. So just a timeline of the research that's been going on, and all the funders there are listed at the bottom. So Aphanomyces was first detected in Alberta in 2013. And then about a year later, in 2014, is when we really started ramping up the research. So we started with surveys looking at how widespread was Aphanomyces. We determined what the oospore threshold levels were, looking at distribution, how the pathogen is distributed throughout the soil. Started lots of field trials looking at seed treatments and cultivars, and also developing DNA diagnostic tools. And that was about over a span of 4 years. And then in 2017, we started looking at cover cropping and different rotational crops that could be of benefit. There's been some challenges with that trial just because of really adverse weather conditions, really over 3 years of the trial. And then we've also been looking at interactions between different pathogens. And then in 2018, as part of the large pulse cluster projects, we've been looking at field trials again, trying to determine what that rotation interval is, what are some pulse crops that we can include in rotation that aren't going to contribute to the Aphanomyces problem, excuse me, and then also looking at intercropping. And then along with that, identification of host defense responses and breeding. And then starting in 2021, we're actually launching a whole bunch of new projects, fusarium cross pathogenicity. I don't know why I'm losing my voice this morning. New and rapid diagnostics to replace, like I said, so that we can look at more what maybe those lower levels are in soil rather than just being able to look at the threshold, liming and calcium amendments, and also these oospore decay models. So now I'm going to go through and sort of talk about what we know in each of these sections and what we still need to determine. So when we're looking at soil tests, what have we learned over the past 7 years? I think the biggest thing was this test series of experiments that really show that the oospore inoculum threshold dose was about 100 oospores per gram of soil. And you can see there in the picture there what those oospores look like and how they get left behind in the roots. We developed DNA diagnostic tools for both Aphanomyces and Fusarium species. I think some of the seed testing labs are kind of launching using that Aphanomyces DNA diagnostic tool, but we're still trying to figure out how to fit Fusarium in quantification into that tool as well. With some of the distribution, we found that inoculum is highest in the top 12 centimeters of the soil. And that's really, if you look at the picture of the roots there, you know, pea roots are generally about 10 to 12 centimeters. And so that's where we see the inoculum is concentrated kind of in that root zone. And then inoculum can be patchy within a field, but it tends to concentrate in low and wet spots. So those are really the target for soil testing. Next slide. So what we don't know, I think what we're really trying to work on now is a more reliable and rapid high throughput method for Aphanomyces and some of the other pathogens, you know, maybe moving to it being able to do in-field diagnostics, although that's challenging from soil. And then the other thing is looking at an integrated tool for both multiple pathogens that also takes into account the soil and environmental conditions. So that's some pretty high level statistics, machine learning, and all sorts of sort of AI computer stuff that I don't know much about, but we're hoping to collaborate with others that have that expertise. So the ultimate goal for this is, if you want to click the animation button, Nevin, really our ultimate goal is to release a decision support system that's based on pathogen quantification, soil properties, and field history, because there's lots of ways that those interact to give a very specific risk for your field. So next slide. So when we're looking at managing diseases, what have we learned in the past 7 years? First, with all our trials with seed treatments, we have found that seed treatments can provide early season suppression, but when you look long-term over the growing season, we really don't see much of an impact. And that's because Aphanomyces and these other root rot pathogens can also come in later in the season, and seed treatments aren't effective past about 4 weeks after seeding. The interval period has been a very interesting challenge because at some locations we've seen pea yields recover after about 4 years, not necessarily disease severity, but the peas will actually do quite well, even with high disease severity. And in other locations, we've been more than 6 years since the last pea crop, and we haven't seen any recovery of yields or disease severity. So that's where this challenge that it's very field specific, and there's maybe some very specific environmental soil conditions that are contributing to how long those interval periods are. We've looked at alternate pulses, particularly faba bean, chick bean, and soybean, and have shown that these are good pulse crops to include in rotation that do not increase risk to Aphanomyces. So they don't get Aphanomyces themselves, and they don't make peas worse. If peas are grown following a faba bean, chick bean, or soybean crop, we don't see the Aphanomyces levels have gotten worse, and in some cases they actually look a little bit better. And then intercropping with mustards has been really interesting, and we're going to expand on that research a little more. We found that the intercropping boosts the pea yield, but again, doesn't have any impact on the root rot severity. So there's something else going on where the peas can yield better despite having these high disease levels. And so that's kind of interesting why that happens, which we don't know why it does. So what we're looking at the next couple years is starting on these calcium or liming treatments to see if they reduce Aphanomyces root rot. And this would be particularly to target some of that patch management. So if you know you have Aphanomyces starting in small areas in your field, can you treat those with lime or other calcium products to prevent spread? We're looking at oospore decay periods, and then two maybe new areas of research that have just started to talk about is this idea of suppressive soils. So we've come across a few soils in the research that no matter how much Aphanomyces we add, they just don't get disease. You don't get disease showing up. So the question is why? And so we want to look at what that microbiome is and to be able to then select, maybe particular good microorganisms that will be adapted and effective biocontrol agents. And then another big question is this idea of what triggers from the host cause the pathogen to germinate or cause the oospores to kind of respond to peas. And if we can figure out what that is, there's this idea, well, can we sort of cause a suicide germination where they germinate and then have nothing to infect? So maybe some new areas of research. And really, this is the holy grail that we want to come up with some sort of disease management besides field avoidance that actually will mitigate root rot. And like I said, right now, that's been one of the most challenging things. Next slide. So when we look at partially resistant cultivars, I always think I'm going to have time, and then I run out of time. So this is work that's mostly been done at the University of Saskatchewan, where they've looked at what the different pathotypes are, Aphanomyces euteiches, present in Alberta and Saskatchewan. And we really need to know that information in order to breed for partially resistant cultivars. And then also at University of Saskatchewan, they've been working on transferring some of the partially resistant lines that were developed in France and the US to Canadian adapted lines for Aphanomyces resistance. I think those are pretty close to the field testing stage, so we'll see how well they hold up in the field. Resistance in my lab, we've been working more on resistance to fusarium species, and that is proving to be quite challenging. There's lots of different species involved, and it seems like if peas have resistance to one, a line has resistance to one species, it's not resistant to the other. So there's this whole interaction of these different pathogens that make it challenging. Next slide. So what we need to know for this, I think the most important question is, if we have these partially resistant cultivars that get released in a few years, how do we protect that resistant source so that we don't lose it? It's taken 25 to 30 years to develop that resistance, to find some resistance, so we don't want to lose it, 5 years after deployment. So to do that, this really requires some more high-level knowledge of pathogen variability, genetics, genomics, how does it infect, all these sorts of more fundamental research questions. And it's the same for how do we find new resistant sources. So that requires knowledge on the host, what are the defense responses, how they're varying between it, how can we develop molecular markers. So again, more of this high-level like lab research that's hard to translate into the field. And then the ultimate goal, of course, is to develop resistant cultivars. Nevin, if you can hit the animation thing. So yeah, we want to release cultivars with resistance against the root rot pathogen complex. So not just Aphanomyces, but we're really chasing the holy grail here of having some resistance to all the root rot pathogens. And then we want to keep that resistance sources protected for as long as we possibly can in the field, which kind of brings us back to the idea of agronomy, disease management, knowing what's in your soil so you know how to deploy these cultivars. So just to wrap up, I think the perspectives for solving root rot, I think everyone that's dealt with this knows it's very challenging for a number of reasons, partly because we're not just dealing with Aphanomyces, we have all these other pathogens thrown in the mix. Foundation, though, is really knowing what's in your soil, and I think that's what I've been chasing in my research, and Holly's going to talk about some of the applied aspects of that and how you can test your soil. And then once we know what's in the soil, then it's building these disease management strategies, which again are difficult. A lot of things that we've tried have not worked, but I think some things, we're starting to see some improvement in some areas. And then of course we want improved resistance or some resistant cultivars out there at least to start with, and then work on continuously improving those. So because this is so challenging, I think we have to start thinking outside the box on some of these solutions, some innovative, creative, and collaborative thinking. And then also there's going to be some more long-term research with this idea of a fundamental research where we're really looking at, well, how does a host become resistant, and how can we exploit some of the pathogen weaknesses to develop management strategies. And I'm sorry I went a little bit over. I said I always get excited talking about research, root rot research, but thanks for listening.
Awesome overview. So happy to have you here today. And I want to reiterate that please, all of the attendees, get your questions in the chat. We're going to go through all three speakers and then open it up for a lively discussion. So thanks so much, Syama. And so if I can multitask here, I'm going to try my best. Just pulling up your slides. Can you see them there, Holly?
Yes, I can.
Awesome. Well, I'm going to introduce Holly Gellech, and she grew up west of Edmonton and was influenced by her father's contribution to farm gate value-added initiatives and international trade relations. She attained her Bachelor of Science in Agriculture from the University of Alberta and has built a career in the seed industry since graduating. Her most recent interests include establishment of a community garden in Strathcona County, and Seeds Canada board member and chair of the Diversity and Inclusion Steering Committee at Seeds Canada. So very warm welcome to you, Holly, and I'll let you take it away.
Most excellent. Thank you, Nevin. You can advance the slides past the one with my image to the following one, and then I'll proceed from there. Yeah, skip past that one. Perfect. So thank you very much, Syama. That was a wonderful presentation. It was very futuristic. I love hearing that there is a pile of research going on in the area of management, in the area of disease resistance. This is excellent. So thank you very much for inviting me to talk today. We are the applied arm and helping out and assisting seed growers, farmer producers on evaluating what in their soil is going to potentially cause issues. So Aphanomyces, we've been conducting testing at our laboratory just outside of Sherwood Park for a number of years now. We understand the issues that come around with Aphanomyces. We understand that in the future, soil health and sustainability is going to be very important, and understanding the soil health. Fertilizer costs is a big thing and an advantage of the pulse industry. Disease management has become difficult with the various types of viruses, bacteria, oospores, you name the different types of diseases that are out there now. They're very expansive. Right now, Western Canada is in a leadership position in the protein industry. We see plants that are emerging, shipping out internationally. This is a huge area and very important to Western Canadian farmers. Protein has become so important to our consumers. They're increasing it in their diets. Lots of discussions around vegetarian diets, flexitarian diets. That's become very, very popular. So we want to ensure in Western Canada, especially in Alberta, that we can maintain our position in the pulse industry. Next slide, Nevin. So the question here that you're asking is, when do I test and how do I test for Aphanomyces? So I've laid out a timeline here for the calendar. Starting in right now is when we're starting to see soil samples enter our laboratory. We start seeing them on Monday. Once the snow is gone, that's when samples start coming in. And the purpose of that is really to do pre-seed planning for your farm. Do you have Aphanomyces? Do you not? How can it be managed? Now, it can also be done in the fall as well as a crop planning preparation, but I do find a lot of the spring samples are, access to new land, maybe lack of familiarity on the land, rental agreements, research organizations. So soil tends to be the preference in testing as you head into your planning for this upcoming seeding season. Now, in the summertime, we do test the tissue. So that starts typically at the end of May, beginning of June, when symptoms start showing up in the field. Typically, you'll see a very even germination, the crops coming up, there's elongation, no development, looks fantastic, and then you start to see the stunting and the browning of the plant above ground. So typically we see tissue sample testing starting at the end of May and producing all the way out till harvest, so all the way out to August. Next slide, Nevin. Okay, so what are you testing? So we test 3 different things in our laboratory, and I'm going to go through each of the different tests that we do offer. So know your susceptible crops, know your field history, and if you don't know your field history because you are renting, please do find out as much as you can about it, as well as know your waterways. That's such an important part. As Syama indicated, the pathogens tends to come in the waterways, so be very keen on those areas. So during the growing season, when you are submitting and getting ready to submit samples to the lab, you're starting to see symptoms in the field. The first thing is to bring out your shovel, head out to the field, and start picking out the samples that you want to ship to the lab. So what we want to see is symptomatic root tissue only. We don't want to see any above ground plant material. So you are taking your shovel, being very careful because with this pathogen, what it really does is it destroys the root. So very careful sampling using a shovel. The next step would be is to bring those root tissues into your shop, clean them off gently with a light water, then let them air dry for roughly 24 hours. And then that's when they are to be submitted to the lab, wrapped in paper towel and sent in a container that's not going to damage them. One of the things that's very important is don't send them in the mail. We've had samples show up that have ended up stuck in a postal service for a week. And by the time they get to us, they are moldy and green and just we can't work with them. So recommendations again is to wrap them in paper towel, ship them by courier, or drop them off at one of our lab locations. We do have another lab in Grand Prairie, so up in the Peace Country, so samples can be dropped off there and we can coordinate overnight courier to our Sherwood Park lab where the testing is conducted. One of the other things is to keep in mind when you're out sampling the root tissue, if this is a new disease for the field, what we'd recommend is also take a soil sample from the same area you're taking your root. And the reason I mention this is it's something that if this is a new development, a new infection, it may not be picked up on the initial test from the lab. So you want to have some backup soil. So when you're soil sampling, as Syama indicated, she said the top 12 centimeters, exactly correct. So you're not looking at, getting deep down at, 12 inches that you do for soil nutrient testing. You are simply looking at that top A horizon, those 3-4 inches. That's where the root is. Now, when you're soil sampling, especially on land, new land to you, you are looking at those waterways. That's the area you need to be in. Now, when you're submitting the sample to the lab, important to submit your sample submission forms and ensure the outside of the bags are clean. And you think, oh, that's kind of a weird comment, Holly, but spring sampling is a very muddy adventure. If anyone, I'm sure each of you have been out there soil sampling for nutrients in the spring, and it's a muddy mess. So we do recommend that you clean the outside of the bags, ensure everything is spick and span. And the reason for that is we want to limit introduction into our laboratory because this pathogen is very important. We need from a lab perspective for samples to come in clean. Next slide, Nevin. One back. Perfect. So how do we test for Aphanomyces? So when we receive a sample, we will take the soil sample or the tissue sample and subsample a portion of it for diagnostics. The tool we use is a PCR DNA test. For soil, we will subsample and weigh out sufficient amount to go into our microtubes. For tissue, we will then chop off a portion of what appears to be the infected tissue, and then we will then get that ready for processing. So under the fume hood, you could see that one of our analysts here, Anu, is preparing the DNA extract. Once she's completed that, it goes into the robot, the picture underneath it, where all of the PCR reagents are added to the specific tubes, and then it goes into DNA analysis in our BioRad PCR machine. And the output is the graphs that you see on the bottom, which will indicate to us whether the disease of Aphanomyces is detected or not detected. Next slide, Nevin. So how we report as either detected or not detected. And in Syama's presentation, she indicated that at 100 spores per gram of soil, that's where the current detection is at. So LOD is limit of detection. So that is where we set as a benchmark. We feel very comfortable that we are able to replicate the result at 100 spores per gram of soil, as well as that's where infection starts to show up if the conditions are right for the disease. So if you do have a detection on the PCR, your next step is, of course, your rotational management and crossing the fingers that there may be other solutions as we move forward. I believe there's strong research on new varieties, management, some of the work that Syama's working on. But if you are very interested in expanding it, is quantification testing. And this is something that's very new to our lab. We are starting to work on it, and that is quantifying the exact spore load per gram of soil. So is that, are you in that just detected mark, say, 100, 120 spores per gram of soil? Or do you have a more moderate or heavy infested soil at 5,000, 10,000 spores per gram of soil? So that is something that we can help out with. And so do contact our lab, as we can do quantification testing. But one of the key things we're going to need to know is your soil type. Are you brown? Are you a brown-black soil type? As we do find that the spores can bind quite tightly to some of the soil types that we have out there. So that's a helpful tool for us. Now, if you get a not detected result, and that will seal your decision. Okay, this looks like something I really want to move into. I'm going to plant my pulse. But then you understand that there's still some unhealthy plants out there, and you're trying to figure out, what can I do next? What else is impacting? Next slide. So if you do have a not detected result for Aphanomyces, and you want to explore further what could be impacting the growth of the plant, we can offer a subcontracted service. This is not conducted in our lab, but it's out in Ontario, what's called a DNA multi-scan. And it's a comprehensive diagnostic tool of both soil and tissue that will cover a broad grouping of pathogens. There is over 60 pathogens in there, because it covers all crops, whether it's cereals, whether it's vegetables, fruits, etc. So there is a number of pathogens in there that are not applicable to pulses, but they do cover Aphanomyces, some of the Fusarium species, Pythium. So that's something that is very helpful to some producers that don't have Aphanomyces but are interested in understanding what's going on. The rating that does occur on the report of analysis is not detected for each individual pathogen, or a low, moderate, or high infestation. So this is an extra tool for those that are very inquisitive going forward. Next slide. And that's it for me. So thank you very much for listening to my conversation on what kind of tools that we do have for both tissue and soil testing. So at this point, I'll leave and close with, if there's any questions, please contact me. Here's my contact information, and I could provide more details on proper sampling techniques, as well as turnarounds and pricing on the testing.
Awesome. Thank you so much, Holly. Very good information on the types of tests that are available and tools that growers have at their hands. So I'm going to take a quick moment to introduce Mike Hilhorst. Mike is a grow team advisor with the Federated Cooperatives. He's worked in the industry for over 20 years, holding roles with various companies such as Dow AgroSciences, Arista Life Science, Farmer's Edge, as well as independent and co-op retail agronomy positions. He is responsible for agronomy, training, sales, and technical support to co-op retails in North Central Alberta and the Peace Country. He's also helped lead 4R nutrient and digital ag initiatives across the cooperative retail system. So Mike and his wife Kelly live southeast of Wetaskiwin on 10 acres and have 3 children and 2 grandchildren. So very welcome to you this morning, Mike, and glad to have you with us. Really interested to know, what changes you've seen over time, what your team is seeing in regards to Aphanomyces and other root rots out there. So I'll hand it over to you for a few minutes, and then we'll ask the other panelists to join us, and we'll take on some more questions. So welcome, Mike.
Yeah, thanks for having me, everyone. Aphanomyces is one of those things that, similar to clubroot. I live in kind of the clubroot epicenter, unfortunately, in central and northern Alberta here. And here again is another disease that's raising havoc, with producers that are, trying to do their best and rotate crops. Generally speaking, we see fairly tight wheat and and canola rotations, but there are, quite a few growers that, do put pulses into their rotation to try and help with that, 4 to 6 year ideal rotation that we want to see with Aphanomyces, and similar to clubroot with canola, the more diverse the cropping system can be, the better off we're going to be. But really from my standpoint, it's something that's here now in central Alberta, even in the Peace Country of Alberta and BC. It's been confirmed last year as well, so it's here, and we have to try and manage it. And it's going to be a challenge, but I think there are some some tips and tricks that I've seen and try to use with producers that will at least set us up for a good start. And really that comes down to number one, the seed that we're putting in the ground. In most cases, we're putting in $250, $300 an acre of fixed and variable costs into every crop that we're growing. So if we're putting weak soldiers into the field, as I like to call them, we're not setting us up for a very good success there. And seed testing is something that I really harp on with retails and producers. Not just a germination test, but vigor tests and a complete disease screen, especially when we're talking about a new disease like Aphanomyces, Fusarium, botrytis, things like that we can see on a seed, full seed disease screen are pretty important, and it helps us make that seed treatment decision. And it may come down to the point where we don't seed that particular lot of seed if we think it has too much disease. But generally speaking, the seed treatments like Trilex and Vibrance and Apron and some of those out there will do a good job of helping control those seed-borne pathogens that may be on the seed if you decide to plant it. From there, soil testing. This is a disease that's very water dependent, and unfortunately, it's something that can affect from seedling to harvest. So it's very, very difficult to control. Even with seed treatments, we're probably looking at 2 to 3 weeks of control. Even with a product like Indigo Solo from New Farm that's has some suppression of Aphanomyces, you're probably only going to see that for 2 or 3 weeks. And something that happens, an infection that happens, or a rainfall event that happens, into that later crop stage can be detrimental to it. So one thing that I've seen in our area especially is we've seen some producers move away from peas and go to crop like faba beans, which seems to have some good tolerance. Far easier to harvest, generally speaking. It likes water, compared to peas. So we've seen some good success with faba beans, though there can be some challenges in seeding faba beans. Generally with the seed size, we have to seed at very high rates. And when I think about the overall rotation on a lot of farms in our area, when we're growing a lot of CPS wheat, when we're growing a lot of canola in rotation, that's kind of putting you to the mid to the end of the harvest window. Then you add faba beans into that without any barley or peas, and you're September, October combining every year. And with the falls we've had over the last 4 or 5 years in a lot of areas, it's been a battle, it's been a struggle. So putting that another late season crop into the rotation can be pretty tough. Another thing is soil testing. We have a lot of areas that have some solonetzic issues, have some salinity issues, and we got to be soil testing not just in those those 0 to 6 inch depths, but down to depth, especially with salinity. We find that there's some hidden salinity in certain areas, and that can affect any type of root growth, especially peas when they get down into that 1 or 2 feet during the year. So that's something to consider. Generally speaking, in central northern Alberta especially, we have very, very low pHs. On a composite test, we have some values coming back at 5.5. So on a composite, there's obviously areas that are probably closer to 5 or below 5. So that's going to affect crops like peas especially, and we have to look at that. Maybe even looking at some zone management of fields as well. Like I said before, we put in $250, $300 an acre of inputs into a crop in areas of some fields that we know they're just not going to perform, and they never have. So I question that sometimes. So I think that we got to start looking at micromanaging our fields more often if we can do that. Most growers have the technology in their controllers and their sprayers to do those kind of things, and we just need to make sure that we're utilizing them as well. As far as seeding goes as well, we have to have a good idea of phosphorus levels in the field. I see a lot of growers kind of skipping their phosphorus application in their pea year, and I question that. I know it's far easier just to fill your drill tanks 1 and 2 with peas and put your granular inoculant in your canola tank, and away you go. hey, I've done it before too. It's way easier, but I question the threat of maybe drawing down your phosphorus levels that much more, in that P year if you're not doing that. So I think we got to look at the soil test, see what we're dealing with, and apply appropriately too. And there is some question as well out there about potentially adding a little bit of nitrogen fertilizer to peas, especially when we have confirmed fields with Aphanomyces. So that's something I guess to think about as well. But at the end of the day, this is going to be a challenging battle, and in some cases too, it just comes down to the field that we're putting peas in as well. If you have more sandier textured soils, or maybe a little lighter soils versus some of our heavy clays and gumbos that we see in a lot of areas in my home area here, maybe we need to avoid some of those fields to put peas in whatsoever. But it's not easy. It's going to be fun, but certainly glad to hear from Syama that there's some potential resistant varieties or potential for that coming down the road. And but at the end of the day, similar to clubroot and canola, we just really have to start thinking about more crops in rotation. I think about some really new things. If you live close to Bruderheim or within an hour of Bruderheim, Alberta, maybe it's throwing a fiber hemp crop in. When I used to work in the Peace Country 20 years ago, there was probably 1 million acres of forage seed production. And there's still some very lucrative contracts from companies like Brett Young and Pick Seed for growers to put some of that grass seed, the tall fescues, the creeping red fescues, back into rotation in the Peace Country, which used to be commonplace. So just a few things that to think about as we go into 2021 and into the future.
Awesome, Mike. Great points. I know certainly you hit home with faba beans being a late season crop. I know on our farm we are trying to bring faba beans and keep them in the rotation, but it's tough to have faba beans earmarked for all of your acres. There's also the question of marketing as well, which can bring producers some difficulty. I'm really glad you hit on fertility and the importance of phosphorus, especially in those fields that might be deficient, because I've seen growers where they've run out of phosphorus and they've finished off the field, and it's like night and day where you can see the difference in colors. But I have a question to you, Mike, and I'd ask the other panelists to turn on your cameras and your microphones. We're going to get to questions to everyone, I'm sure. But Mike, how important is it to manage the entrance to fields? You mentioned forages, etc., being a potential viable solution to at least breaking up the years out of susceptible pea or lentil crops. But what about managing approaches? I know we have lots of compaction. I know that Aphanomyces and clubroot both tend to see symptoms in those areas. Do you have any growers or any I guess suggestions on how growers might better manage those approaches?
Yeah, I think living in Clubroot Alley here, I think it's been fairly commonplace for most growers over the years, and custom applicators especially, to when you're moving from field to field or from farm to farm, to to knock off that loose dirt and debris off the machines. I think that can go a long way in helping with the spread of Aphanomyces, and it certainly has with clubroot. But everybody has to do it, and I don't necessarily see that all the time. There may be some farms that do a fantastic job, and then someone comes ripping down the road with mud flying off their sprayer tires, and it's a little disappointing to see that. But at the same time, it's not easy to stop for half an hour when you're seeding, or if you're in the middle of spraying, to to stop and do that. I understand that, but it really needs to happen to help stop the spread of some of these things, from a farm to farm level. Seed moves from province to province. Equipment sales, equipment moves from province to province. Is it always 100% clean or washed down, or maybe even to the point of disinfected? In a lot of cases it isn't. So it's challenging, but really kind of comes down to compliance, I guess, if you want to call it that, and everybody kind of working together to to mitigate the spread. Really is what what we're trying to do. So
excellent. So I know that we have some questions that are coming in over the chat, but I'm going to get the first one in. I have a question, Holly. You mentioned the importance of testing in regards to better preparing producers, making decisions on rotation, which crops. How much do the tests cost?
Oh, that's a really good question. So whether it is a soil or a tissue, it's the exact same processes that are in our lab. So it's going to cost you around, for our lab anyways, is $108 plus tax.
Excellent. Good to know. A small price to pay to having that knowledge and that arrow in your quiver, so to speak. And Syama, I have one more question for you before I turn it over to you panelists to perhaps ask each other a question. I think, you know, we saw that most of the producers here in the audience are growing peas and lentils. Some are growing other pulse crops. What you mentioned, soybeans and chickpeas being very resistant. What's happening with dry beans in southern Alberta?
Asking the tough questions, Nevin. Dry beans, generally the root rots, when we look for root rots, they actually look really good. Their roots are a little woodier than peas, so they can hold up, stand up a little bit better, a little sandier soils. Now, having said that, we have had kind of our first positive detection of Aphanomyces on dry beans down in southern Alberta in a couple of fields. We are still trying to, there's a whole bunch of processes that as scientists we have to go through to confirm that a pathogen is indeed a pathogen. So we're still trying to get through all those steps. The whole COVID shutdown in the lab kind of prevented us from doing the very last step, which is basically sequencing some genes from that pathogen to make sure that they exactly match Aphanomyces. So we haven't completed that. But yeah, generally like the crops look really good. It's just a couple fields where we have found Aphanomyces, tighter rotations on these fields. So that's probably at play. Dry beans is a little interesting because of all the different market classes, there is some differences in susceptibility resistance of the different market classes as well. So that's something that we want to explore. And we had plans to do that in 2020, but he said everything was kind of put on pause. So, you know, there's lots of questions about dry beans, but I think in general, it can be a risk to the industry, but we're not seeing it to the same extent that we've seen in peas. And then just one last thing is that Aphanomyces that can infect pea appears not to infect dry bean and vice versa. So it's like a separate, dry bean have their own strain and pea have their own strain. So it's just interesting that it's kind of popped up when it's not, pea's not actually host.
Excellent, thank you. Another question probably for yourself or perhaps for Holly is coming in from Doug Sell near Bicycle, Alberta today. At 100 grams of soil, does the spore load at this stage affect yield? So that would be yeah, I think that's how I got it right.
Yes, it does. And I don't, like I couldn't put up too many pictures today just because of what I covered, but it's an interesting pathogen in that it's like plus or minus. You up into 100, you really don't see any disease at all, and then you hit 100, and then all of a sudden it's just like the switch gets turned on, and you see a lot of disease. So even at 100, you know, we're looking at fairly, the roots get very rotted, fairly decayed, pretty much up to kind of our highest disease severity level. So yes, we do see some yield loss. What's been very challenging, I think the message of all this is that it's just challenging. We've been trying to develop sort of these yield loss models where, you know, where you look at, okay, if you have this certain amount in your soil, can you predict what your yield loss is going to be? And the graphs, there's no nice correlation or nice, you know, usually you want to see a nice line, and we just see it all over the place. So it's really dependent on environmental factors, site-specific factors. Like we have one of our sites up at Lacombe that's probably at about 100 to 500 oospores per gram of soil, and we pulled off yields of like 4,000 kilograms per hectare. And then our site down in Tabor, which is very similar oospore levels, we get 1,000 kilograms per hectare. So, you know, you're just like, well, what's going on that you see this huge discrepancy? Sorry,
yeah,
my answers are always complicated.
Well, it is though. I mean, it's a tough correlation, right? I mean, we have variable soils, variable moisture throughout the soil in every field. You know, it's going back to some of that zone management that I talked about in fertility, and the same thing goes with you know, disease like this. There's areas where it's going to be awful, and there'll be other areas where those peas will be 80 bushels an acre. It's a tough deal.
Mm-hmm.
Excellent. Do any of the panelists have questions or to each other or any comments? No?
Nevin, can I ask a question?
Yes, I hear Jen Walker. Please go ahead.
Good morning, everybody. I actually have a whole bunch of sticky notes of questions. So unless anybody else has something, you know, if you're listening in, please put up your hand. Nevin, I'll give you some time to read Mark Olson's question. It's long and complicated, but this is kind of for everybody, and I'd like everybody's perspective on it. Mike, you touched on it. Holly, you mentioned it. Syama, I know you and I have had conversations about it. But traditionally, when I as a farmer go into a field, I manage fence to fence. And with diseases like Aphanomyces, Mike, you mentioned clubroot, and I know you've got some great experience with clubroot and liming and those kind of things. Is patch management feasible on a whole farm level? And how important is kind of, you know, technology? Mike, you mentioned variable rate. Holly, you talked about waterways. And I think in the last couple of seasons when we had some really soggy years, you know, we can see that tracking. How important is it that we shift our thinking as farmers from managing a field fence to fence to looking at kind of this more zone management idea? And how is that going to impact not just Aphanomyces, but I think other diseases as we see kind of, I guess kind of it's really an evolution, right? The more frequent crops grow, the more we're going to get diseases. I don't know who wants to take a stab at it first.
I can start.
take a stab at it. From a zone management, it can become very challenging, but we have technology and we have tools now to do it. Farms are getting bigger, absolutely, but along with covering more acres, they also have more staff, better internet, amazing opportunities to manage variable rate. So I don't see why we cannot manage some of these pathogens in patches. Now, each field is different, especially those ones that have a lot of hills up and down and up and down. We know those areas. Those will be more challenging, absolutely. But I think that as we move forward with even sustainability, management of fertility will become very, very important. We see this in Ontario. We have a seed lab that does soil nutrient testing in Ontario, and you know the 4 Ns are so important in managing what we're doing and more on a micro basis. Even though we're getting bigger, the farms are getting bigger. We're gonna need to adapt, and it will be an evolution. But I think it's coming.
Mike, you had some comments there as well.
Yeah, you know, I think from my perspective, I think about liming, and you know that could be one of the most beneficial things we could do in many areas of the prairies. You know, we've got very low soil pHs, and it's inhibiting plant growth, it's inhibiting phosphorus availability tremendously, and you know the biggest challenge right now is cost and logistics of lime. But I mean, if we try and do it on a zone level based on pH mapping and things like that, I think, you know, there's probably 20–30% of a lot of fields that actually need it. That would make a tremendous, you know, would make have a tremendous benefit on doing that field. And I think about a neighbor of ours, both Jen and ours, Scott Keller, who grows a lot of pulses in his rotation. He started to do some liming, some variable rate liming, and you know, when you look at the cost, the other thing is the investment is big, but you got to think about it long term because it will last a long time as well. In some cases decades, if you're putting enough lime on. But the challenges as well is that investment, and you know, a lot of our farms are renting a lot of land. They don't own their full acreage base. So you know, there has to be some discussions, I think, with the landlord long term, with possibly even government to help, you know, mitigate some of these soil issues that are in a lot of cases are the overarching issues to productivity, you know. And liming and pH being one of the big ones that I've seen, and one of the biggest opportunities if we can just try and figure it out. And it's not easy because it's tough to apply. You know, the sources come from a long way in a lot of cases, and you know, but maybe we need to look at things like, you know, beet lime and things like that. I mean, you know, I'm on the lime steering committee for Alberta, and so I get to be part of some of those meetings and some of the research that's going on as well. But as far as liming goes, I think that's one of the biggest opportunities that would help with a whole bunch of different challenges that we're dealing with on the farms these days. So
awesome, Mike. We've got some more questions coming in, and we've got about 6 minutes left. So on the question of liming, beet liming, other types of lime, Syama, I know we have some research in the works. What is the actual pathway that liming is? How does it work? Is it the calcium? Is it the chloride? Is it the pH? And you got 30 seconds.
Good question. For clubroot, it's pH. For Aphanomyces, it's calcium. With the benefit of, adjusting your pH. So why the calcium, I think, is a good question. But yeah, so 30, was that a good 30-second answer?
Got 10 more. So it is something to do with the calcium, and is it the germination of oospores or the pathway of some sort?
Well, the calcium looks like it interferes both with the oospore germination process and then also the zoospores that track and home into the roots, and then they encyst, so it interferes with that. And so calcium is very important in all our biological processes. So the pathogens need calcium to perform these functions, but if they have too much, then it kind of like overwhelms their system, and then they don't know what to do, so it disrupts it that way. So it's getting that like sweet spot of too much calcium that can be beneficial.
Excellent. And a question maybe to you, Mike, or to you, Syama. What are we talking about for metric tons per acre? Are we talking 2, 4? How many tons of lime or beet lime, 0 tons? What do we know about rates? We know the economics of trucking and getting it there is probably the biggest factor, but what are the rates for liming?
Yeah, in some cases it's a minimum of 4 metric tons up to 12 in some areas, that are, quite highly acidic. And yeah, it's a lot, I guess I could say in a nutshell. It's a lot, that if you've ever seen an applicator go across those areas, it looks like a snowstorm. It's and even that, I mean, you don't want to be applying this on a windy day. It's got to be calm, but maybe not too calm, so it doesn't hang for 4 hours in the field. We've seen that with certain lime sources as well. So like I said, it's challenging, but yeah, we've seen 4 to 12 metric ton rates having to be applied to be effective. And for what I've seen anyway, Syama, maybe you can comment on that as well. But
yeah, no, that's exactly it. And the field trials that we're going to start on, those are the rates. We're looking at 4, 8, and 12. It might be 13 tons per hectare. In the greenhouse, we saw that highest rate really it eliminated any Aphanomyces at all, and we didn't get quite the same suppression at 4, but we did get some. And it's going to depend on your field, and it's difficult, for pH. If you're looking at clubroot and increasing pH, then there's all sorts of equations where you can say, okay, this is my target pH, and I need to apply this much lime. When we're looking at the calcium effect, and we don't quite know what that calcium concentration is, and there's lots of different forms of calcium, we don't quite have that target where we're saying, okay, we have to reach this concentration of calcium to have an effect. So there might be some other more detailed sort of mechanistic research that needs to happen rather than just field trials and seeing if it works or not.
Thank you. We're down to the last few minutes here. There's a question in the chat about breeding resistance, etc., to some of the other root rots. I think we're going to leave that one. Maybe we can get back to you via email on that question. Mark Olson has some comments in here in regards to research coming out of Saskatchewan looking at NP and sulfur. And he notes that by far the most important nutrient for yield and to a certain extent seed protein is phosphorus, as Mike, you had indicated. Adding N and S had very little effect on yield and seed protein, but what about the value of adding N? And what about for Aphanomyces? Do we know anything in that regard yet?
We tried some field trials where we did add N, up to, I think it was 80 pounds per acre. Thinking, well, there's no nodulation. If we add N, then do you increase? And we really didn't see any effect on the nitrogen. I think part of the problem is the roots are so decayed, they can't quite take up the nitrogen. And then Nevin, I know we've had conversations about phosphorus, and we are planning on including that in our field trials as well, just to kind of get that, direct side-by-side comparison.
And potassium as well?
Yes, yeah, potassium as well, which I think would function very similar to calcium. But in this case, we'd be looking at putting potassium like right in the seed bed rather than this kind of blanket application.
Excellent. And Mike, question to you with the agronomy services that you provide and are aware of. We've heard some anecdotal evidence that producers using K in the seed row are having good results and are fending off Aphanomyces. Have you seen any of this or heard anything about that?
Not in particular. I've heard, a few rumblings about it. Obviously, potassium, if it's limiting, is a really important nutrient in water transport and things like that, which we're talking about big time when it comes to Aphanomyces and soil moisture and whatnot. But haven't heard anything or seen anything in particular. But it's like any of the nutrients. If it's low and you're deficient, you need to soil test to find out what you're dealing with. You got to see where the gauge is before you start the truck up, kind of thing. Mm-hmm. And if we don't really have a clue, then we may be over or under applying or not applying something that we truly need to to have that plant be as healthy as possible to try and fend off Mother Nature and everything that she's going to throw at us during the season. So
awesome. Well, we still have a bunch of unanswered questions in the question box, so we've had great participation today from our audience. I certainly want to thank each of you individually for your contribution this morning to the first ever APG Tailgate Talk. It was very informative. I learned some new things today as well. Again, I want to thank as well Dr. Jen Walker, Dr. Jagrup Gil-Kaloon, and Rachel Peterson for being behind the scenes and making sure that everything ran smoothly. So again, to all of our attendees and to our panelists, thank you today for a very successful APG Tailgate Talk on Aphanomyces and other root rots. So we hope you enjoyed, and for those who may have joined us late or had to leave early, we will be providing a recording of this on our website. So again, thanks to all of you for joining us today.
Thank you.
Take care.
Thank you.